Okay, let's keep going. So another kind of water movement is called filtration. Filtration happens in your kidney. Your kidney filter your blood and become your urine. So your urine is the waste particle, waste solution come from your blood. That's why we can take your urine sample and find any drug, any medicines exist in your blood. And that process is filtration. So filtration is you have a small blood vessel called arterial, afferent arterial will send the blood to the kidney, and kidney have a lot of small filter called nephron. This is a nephron. And then this is the capillary bed called glomerulus. And in the glomerulus, because capillary is a leaky area, water, not water, blood keep going in. So it's like you push water into a balloon, the hydrostatic pressure, that's the pressure inside the water, will push blood out. So blood's being filtered out. Once it's been filtered out, it will go through the nephron tube. And eventually, it will show up in the bladder, become your urine. So that's how your blood turn into urine. That's the filtration process. And if you want to know the detail, you have to take AP2, the renal system. They will talk into the detail. So in the kidney, that's the filtration process happens to turn the blood into urine. So your urine directly comes from blood. Okay, the focus of this second part of the lecture is those molecules, they could not free move through the cell membrane. They need helper. So we call them carrier-mediated transport. And there are two categories. The first one is called the facilitated diffusion. The second one is active transport. So in the second part of this lecture, we're going to focus on those molecules that could not freely go through the cell membrane. And they need helper, those membrane proteins. So we have a group of proteins. They help the molecules to move, like they help glucose to move from high glucose to the low glucose area. We call them facilitated diffusion because they need helper, membrane protein, transporter protein. The protein we'll talk about in the last lecture, transporter protein. They move the molecule from high to low, so they still go down the concentration gradient from high to low. We call them facilitated diffusion. So we have two examples. The first example is ion channel. So ions move through the ion channel. The second example, they're called carrier proteins, like glucose carrier, and also move amino acid. They move the glucose from high glucose to low glucose, or high amino acid to low amino acid. And that's called facilitated diffusion because they still move the molecule from high to low. So the cell does not need to use energy to make it happen. However, in some situation, you have to move the molecule from low to high. From low to high, like the inside the cell is low. Okay, you want to move it high. And it won't naturally happen. It's like you purchase a pump to move water from your basement out. You know, naturally water won't move out, water will move into your basement. So you need to use energy to make it happen because you are working against nature, against its concentration gradient. And for the biological system, the energy source is ATP. So we call this active transport. You need to use ATP to make this transport happens, move the molecule from low to high. Like inside, if you have hydrogen, when you have too much hydrogen, it will change the pH. So even though the outside has more hydrogen, you still need to pump the hydrogen out. So you're working against the concentration gradient. That's why you need to use ATP to make this happen. So that's active transport. And active transport, several examples. We talk about hydrogen pump, or you can move glucose against its concentration gradient. If it's moving against its concentration gradient, it's active transport. Or calcium pump. All this, they are active transport. You're moving the molecule. The key is moving the molecule from low to high. You're moving against its concentration gradient. It won't naturally happen. You need active transport. And one example, you can click this, watch this video. You talk about the sodium potassium pump. Sodium potassium pump, every living cell have it. It uses ATP as an energy source to move sodium, out of the cell, move potassium into the cell. So it's keep running. So eventually inside has more potassium. The ICF have more potassium. The ECF outside actually have more sodium. And then neurons are the cells in your brain. They can take advantage of it. They can make the sodium and potassium start to flow. And when they start to flow, they generate electrical signal. And that's called the action potential. When you take AP1, you will learn how the neurons generate action potential. So they take advantage of those active transport. It's an active process. So active transport is an active process because you are moving the molecule from low to high. It won't naturally happen. So you need ATP to make this event happen. If you're moving the molecule from high to low, that's called facilitated diffusion. You don't need to use ATP to make this event happen because they just follow its concentration gradient. Okay, and there are other kind of vascular transport. Vascular transport is the cell membrane form a bubble called a vesicle, and they can move the molecule in or out. So if you move the molecule in, it's called endocytosis. If you move it out, it's called exocytosis. And the endocytosis, this is endocytosis, moving in, can be divided into three: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So let's look at each one of them. So all three of them are endocytosis. First one, phagocytosis, also called the cell eating. The cell eat, like your white blood cell, it can swallow a bacteria in. It's phagocytosis. It's a big bite, swallow in, it's phagocytosis. So like this example, the white blood cells swallow in the microbe or other particles, and once they swallow in the bacteria, they can use the lysosome. That's the organelles like the stomach in our body. Inside you have digestive enzymes. You can digest those bacteria. So that's phagocytosis. The next one called pinocytosis. Pinocytosis also called the cell drinking. Cell drinking, because the molecule you move in is either water or liquid, and also it's a small bite. It's smaller, so that's pinocytosis. So there's a picture show you the pinocytosis. They form a small vesicle and swallow the molecule in. The third one is called receptor-mediated endocytosis. So apparently the receptor, when it binds with the substrate, and it will cause the endocytosis process to happen. And the textbook example, receptor-mediated endocytosis, the textbook use HIV as an example. HIV virus, it comes from this virus. It won't affect every kind of cell. It actually will affect the T lymphocyte. Is one of the white blood cell in the lymphatic system, T lymphocyte. So when the HIV virus go into the body, it won't attack the brain, it won't attack the skin, it will only attack the T lymphocyte, because T lymphocyte has the HIV receptor. So when it binds with the HIV receptor, will trigger the receptor-mediated endocytosis. So now their DNA move in and integrate with the lymphocytes DNA. So now we call this person's HIV positive. And the DNA can stay there for years. So that's why the HIV is dangerous, because the host, the person get HIV, and they may not even know they have HIV because they're pretty healthy. So they can transmit the disease to other people. And FYI, HIV actually come from chimp. So in chimp, because we talk about the chimp's DNA, is very similar to human DNA, like 99% identical. So the virus in chimp is not a very serious virus. I just live with them for decades, for 32,000 years actually. And when in 20th century, when humans explore our territory and human and chimp start to fight, so the chimp bite. When they bite, the virus go into human's body. And because humans have very similar DNA to chimp, so when the virus, SIV, go to human body, they mutate. And when they mutate, they become HIV. And those HIV virus, their host is the T lymphocyte. They can trigger receptor-mediated endocytosis. So they only attack the T lymphocyte. And when you don't have T lymphocyte is a white blood cell. So its job is like the soldier, do the fighting. And when your body don't have enough T lymphocyte to do the fighting, a small infection can make you sick or kill you. And that's AIDS, acquired immune deficiency syndrome. Okay, so that's the story of receptor-mediated endocytosis. Another example is the LDL, low-density lipoprotein molecule. It's a lipoprotein. It's a molecule carried by your blood. So it's actually a protein. Their job is to carry cholesterol. So cholesterol will be packed. If they are low density, it's called a LDL, low-density lipoprotein particles. And when they are transported by blood, when you have too much of this, your blood the traffic will be bad. That's why we say LDL is bad. When you do your annual health check, they will check your LDL level. So you want this to be low. But here we talk about the receptor-mediated endocytosis. So when this molecule go to your cell need to take them in. Apparently, you have the receptor, and they bind with this LDL and will trigger the receptor-mediated endocytosis. So now this LDL will go into the cell. Those cholesterol go into the cell. That's another example of receptor-mediated endocytosis. So endocytosis, we talk about 3 examples: phagocytosis, pinocytosis, and receptor-mediated endocytosis. They all need to use ATP, so they're active transport as well. And the last one is exocytosis. Exocytosis is reverse of endocytosis. Endocytosis is moving in. Exocytosis is moving out. So this time you have a vesicle and you gradually move out. So the particle inside the vesicle and will be released. Like endocrine cells, your endocrine cells release hormones, and your neurons are the cells in your brain that release neurotransmitter. And they use the exocytosis process, so they can release those peptide hormones. And because there are 2 kinds of hormones, there are steroid hormones, goes through a different mechanism, and this is a peptide hormone, so it goes through exocytosis. And the neurotransmitter being released by neurons, and it goes through exocytosis. So they also need to use ATP. So they belong to active transport. So we talk about those molecules. They could not directly go through the cell membrane if they need helper. So if they move from high to low, they follow the concentration gradient. We call them facilitated diffusion. And we talk about sodium channel. We talk about glucose carrier or amino acid carrier. And if you're moving the molecule from low to high, it's called active transport. Active transport, we use sodium potassium pump and hydrogen pump as example. Then we talk about other kind of active transport. They also need to use ATP, like the endocytosis, exocytosis. And endocytosis can be divided into 3: phagocytosis, pinocytosis, and receptor-mediated endocytosis, which we use the HIV virus and the LDL particle as example. And exocytosis, that's when the neurons and endocrine cells release neurotransmitter and hormones. Okay, let's stop here.